Welcome everyone. Today we're going to learn about a very interesting topic. But before that, I want to ask you a question that have you ever wondered how our body grows, or how our wound heals, or how our body operates like a well-functioning machine? Well, that is because of mitosis. And that is the topic of today's video. Now, let's imagine that somehow you got the ability to duplicate yourself. So suppose that this pen is you. So 1 second there's one of you, and the next moment, boom, there's two of you, another identical version of yourself. Now this exact thing is happening inside of your body, and that is thanks to cell division. Now we know that cell division, there are two major types of cell division. One is mitosis that we're going to talk about today, and another one is meiosis that we're going to cover in another video. So the question is, what is mitosis? We know that cells are the basic functional unit of life, and mitosis is the process that divides the cell into two identical cells. Now, when I say identical, I mean everything is similar between the mother cell and the daughter cell. The mother cell is the one that is being divided into two daughter cells. So everything, I mean the structural characteristic, the physical characteristic, everything will be similar to that of the mother cell. And by everything, it also includes the chromosome numbers. So suppose the mother cell has 4 chromosomes, the daughter cells, the two daughter cells, will also have 4 chromosomes. So we can sense that this mitosis process is a well-organized, very complex process. I mean, obviously. So this process is classified or divided into these 5 phases. So our first phase here is prophase. Okay? So prophase is the phase where the story begins. So here we have our cell, and it mainly focuses on the genetic division of the genetic materials. So within a cell, we know that each cell has a nucleus. This is our nucleus. The red circle is our nucleus. And within nucleus, we know genetic materials exist. So like DNA. I know some of you may confuse this genetic terminology of genetic materials, such as some may say that it's DNA, some may say it's chromatin, some may say it's chromosomes. So which is which, right? So we know that DNA is the building block of life. So suppose this is our DNA. Okay, this is our DNA. This DNA will coil up, they will wrap around and form this chromatin, this thread-like, thin thread-like green structure. This is our chromatin, okay? So this chromatin will further condense into a chromosome. This condensed version of chromatin is this chromosome. So in this phase, what happened is that the chromatin, they were just chilling, they were minding their own business, okay? So they will all of a sudden condense into a chromosome. Here we have our chromosome. This chromosome has 2 identical arms, which are referred to as chromatid, and they are attached to each other at the center with a centromere. So that is our phase 1. And in the phase 2, we have prometaphase. The name of the phase 2 is prometaphase. And in this phase, we're going to introduce a new character. So the character is the spindle apparatus. The spindle apparatus have this spindle fibers. Okay? Spindle fibers. And at the center of this spindle apparatus, this middle section, we have our equator. So this black dotted line is the equator. And this is the spindle fiber. So what's going to happen now is that this chromosome, they were minding their own business as chromatins, right? So now they're like, okay, we didn't ask for this, so we got to take some action. So they're going to come, these chromosomes are going to come and bind themselves to these spindle fibers. So these chromosomes will come and they will bind themselves to these spindle fibers with the help of the centromeres. They will bind themselves to these spindle fibers with the help of the centromeres. And with that, we can also notice that the nuclear membrane, we know that nucleus have an outer membrane, right? So the nuclear membrane, here we can see that the nuclear membrane and the nucleolus, are like, okay, we don't want to be a part of this, so we're going to leave for the time being. Just give us a call and we're going to come back. So the nuclear membrane and the nucleolus at this phase will start to disappear. So this whole thing that this is occurring in the phase 2, which is the prometaphase phase. Next, we're going to go to the third phase, which is called metaphase. Now this is a very interesting phase because it is the phase where the lineup is going to take place. So what's going to happen is that all the chromosomes that have bound to the spindle fibers, they're going to align themselves at the equator line. So this is the equator line, right? We know that this is the equator line, and the chromosomes are now at the center of the cell, this spindle apparatus. However, we can also notice that the chromatids of each chromosome have their arms extended to the different sides. This is because of the poles at the end. Now this is not the North Pole or South Pole, okay? These poles will attract these extended chromatids to the each different poles, which is why they are extending their arms to different sides. Now at this stage, the nuclear membrane and nucleolus is completely gone. And also, another interesting fact is that during this phase, the chromosomes will become much shorter and much thicker. So it will become shorter and thicker. So this is an interesting characteristic that helps us to differentiate this phase from all the other phases. So after that, our fourth phase comes, which is the anaphase phase. So in this phase, this is like the climax of the whole process. In this phase, what will happen is that the chromatids will separate from each chromosomes. So now each chromatids have become a new chromosome itself. So the chromatids will separate and race to the different poles. You see the poles have attracted them and they are racing to the different poles. So they are not in the equator line any longer. They have reached the opposite poles. Now here's an interesting fact that I said earlier that everything will be similar in between the daughter cell and the mother cell, even the chromosome number. So you can see here that each chromatid has moved to the different poles. So when the cell is going to divide, there is going to be 4 chromosomes at this side and 4 chromosomes at this side as well. This is why we call this mitosis process an equational division, because everything is equal. Even the chromosome numbers are equal, everything is similar. And also when the chromatids are moving to the different poles, they can take different shapes. So there are 4 different shapes, which are V, L, I, and V, L, J, and I. And each one have their own name: metacentric, submetacentric, acrocentric, telocentric. So this is our 4th phase, and then comes our last phase, which is telophase. And this phase is basically just prophase but in reverse. So in this case, what will happen is that the chromosomes will start to thin again. They will go back to their old self, which is the chromatin. They will exist as chromatin again, and the nuclear membrane that decided to leave earlier, they are going to come back. So the nuclear membrane and the nucleolus will reappear. Okay, so that is how, or at the end, there we have our 2 daughter nucleus. So we can see that there is one nucleus here and another nucleus here, and they have been separated. So this whole, up to this point, this whole process was the division of the nucleus of the cell, and this process we will refer to as karyokinesis. So the division of nucleus is referred to as karyokinesis. So this is going to be nuclear division. However, we know that the cell also has a cytoplasm outside the nucleus. This is our cytoplasm. So if we were to divide the whole cell, we would also have to divide this cytoplasm as well. So after the division of nucleus, cytokinesis takes place. So what happens in cytokinesis is that the cytokinesis refers to the division of this cytoplasm. So in this case, for plant cells, the cytoplasm will be divided with the formation of a cell plate. You can see at the middle there's a cell plate, and this cell plate will divide this whole cell into 2 daughter cells, which are identical to their mother cells. So there is one cell here and one cell here. They will be separated. And in the case of animal cells, we can see that there's a contractile ring at the middle of the cell, right? So this contractile ring, what will happen is that they will form a cleavage furrow. This, you can see that they're in the middle, they're moving towards each other, right? The ends are moving towards each other, and so this is referred to as a cleavage furrow. This cleavage furrow will cleave the cells into 2 daughter cells. Here you can see that the cell has now divided completely with its cytoplasm and the genetic material as well, and as a result we have our daughter cells. So from the mother cell we now have identical 2 daughter cells. So next time when you have a paper cut or whatever cut you have, or when you are seeing a plant grow, you can always remember that the cells are always at work with the help of mitosis. It's mitosis that preserving your life and helping you to grow. So that was all for today's video, and thank you. Stay tuned for the next video.